A recently emerged bat-borne zoonotic agent which is highly lethal to horses and humans was first identified in 1994. At the end of 2010, there had been 14 recorded spillover events which resulted in the death of 44 horses and 4 humans in Queensland and adjacent northern New South Wales. The human equine case fertility rates are 57% and 75% respectively. The virus's main hosts are flying foxes. These include the black flying fox, little red flying fox, speckled flying fox, and the grey-headed flying fox. These 4 species of flying foxes have been all affected by natural vegetation clearing and have increased their reliance on their vegetation in urban and exurban areas. The urbanisation which has been caused has potentially driven an increase in pathogen transmission to domestic, animal, and human hosts. The specific way of transmission of the virus to horses and human from bats is unknown, although ingestion by the horse of bat excrement, partially eaten food, or reproductive fluids is plausible. The virus can survive for more than 4 days in the urine of a flying fox if it is 22 degrees Celsius, but is inactivated in less than 24 hours if the urine or fruit juice in which it ingests is 37 degrees Celsius. Seasonal and climatic factors may also contribute to the spread of pathogens in the environment and define the period of time in which the indirect contact may occur. Although Hendra virus is potentially present continuously in coastal Queensland of northern New South Wales environments, although will vary in the infection load. The Hendra virus spillover from 1994 to 2010 was dispersed along a 1,500 kilometre of the eastern coast of Australia. From 1994 to 2010, 85% of the D spillover events occurred in the dry season of May to October. These months recorded the lowest temperature and rainfall for the spillover sites. The mean temperature of which the spillover sites occurred was 17.7 degrees Celsius and 31.8 degrees Celsius, and the rainfall was 0.2 to 237 millilitres. There was also between 13 and 30 days within the months when the spillover events occurred. The increase in frequency of the spillover events is not associated with the climatic trend, as half of the events were less than equal to, or the median of the rainfall to the year, month of the event and the temperature of the month at which it occurred. The location of the spillovers dispersed along a 1,500 kilometre range along the eastern coast of Australia. The locations are within a 20 kilometre radius, an average range of a nightly feeding of a flying fox. 10 spillover events occurred also when a 20 kilometre of a permanent roost of the flying foxes. The study conducted by Rosemary McFarlane, Niels Becker and Hume Field demonstrates a large association with the dry season of spillover events of Hendra viruses between 1994 and 2010. A constant pattern of lower rainfall temperature and primary productivity are connected in all the sites between the months of May to October. Although the sites where the Hendra virus emerged were very significantly in latitude, climate, biography, human and equine populations.